The typical prokaryote has somewhere between 1,500 to 7,500 genes, but they have a problem: limited resources. They don't have enough amino acids to express all those genes at the same time. They don't have enough DNA, they don't have enough RNA nucleotides, they don't have the resources. They don't have enough ATP. So how do they control gene expression so that the right genes are expressed when they're needed and genes that aren't needed aren't expressed? We'll take a look at that in this video. Your objectives for this video are to distinguish between constitutive and non-constitutive gene expression, to label the parts of an operon and state their functions, and to describe repressible, inducible, and catabolite repression operons. So at the beginning of this video, I posed a question: how does a bacterium keep from expressing all of its genes at one time? It doesn't need all of the genes in its genome at one time. For example, a bacterium may have genes to produce the right enzymes to break down specific carbohydrates like arabinose or lactose. But if those carbohydrates aren't in the environment, producing the enzymes to digest them would be a waste of resources. Bacteria, like other organisms, control their gene expression through transcription control. You see, prokaryotes have two types of genes. They have the constitutive genes and the non-constitutive genes. Constitutive genes are always on. So when we say on, we mean that they're being expressed. So constitutive genes are always expressed, aka they are always on, but they're expressed at a fixed rate. Examples of constitutive genes would include those genes that are essential to living. These are sometimes called housekeeping genes. They might be genes that encode cell replication proteins, or genes that are related to maintaining the cell wall, or genes that are related to DNA synthesis and transcription, genes that are related to producing proteins that make ATP from nutrients. Non-constitutive genes are genes that are expressed only as needed for growth. And there are three categories for this. You have what are called the repressible genes. They're always on, but can be turned off. You have inducible genes that are always off, but can be turned on. And then you have catabolite repression, which is sometimes called the glucose effect. This type of repression is on as long as there's glucose around, but as soon as glucose is all used up, the repression stops and the genes get induced. So we're going to talk about all three of these. The first type of operon that we're going to talk about will be the inducible. But first, let's talk about transcription, and then we'll talk about how the operon is laid out. Now, you've probably been through this before, but I just want to be sure that we're all on the same page. Transcription is the process whereby a DNA gene gets read by an enzyme called RNA polymerase. And what RNA polymerase does is it reads the nucleotides, that's the letters in purple here, it reads the nucleotides of a DNA gene, and then it adds the RNA complementary nucleotide opposite. So where the enzyme sees an A, it's going to insert a U, because remember, uracil is the partner to adenine in RNA. There's no thymine. So wherever A is seen, it adds a U. Wherever the enzyme sees a C, it adds a G, and vice versa. Transcription always starts at a particular site at the beginning of a gene called a promoter. Promoter sequences have a specific sequence of A's, T's, or A's, T's, C's, and G's that is recognized by special proteins called transcription factors, and then RNA polymerase can bind at that site. So once RNA polymerase binds at the promoter, It starts reading nucleotides and then inserts the RNA base opposite, and this is how RNA gets made. So here we see an transcription bubble being formed as RNA polymerase opens up the DNA double strand, then it starts reading sequence and making RNA, and it continues doing this all the way down until it gets to the end of the gene. At the end of a gene, there's a special sequence called a terminator sequence, and transcription stops at that sequence. The RNA polymerase falls off, or it lets go of the DNA, and transcription ends. So an operon is basically a transcription cassette, okay, a functional cassette, and it's made up of a few important components. For your exam, you should be able to state the function of the different components that I'm getting ready to talk about. The first part of the operon is called the control region, and it's located basically here. In the control region, there are two sequences that you need to be familiar with. The first is the promoter sequence. So remember, this is where RNA synthesis starts. The second site is called an operator site. So this is supposed to say RNA synthesis. The second site is called an operator site. So operator site is where transcription control is going to happen. There's a protein that is encoded by this regulatory gene that's just outside of the operon. The protein is called a repressor, and it has the ability to turn off gene expression. The repressor protein is able to bind the operon here at the operator site. If the repressor protein binds there, then that means when RNA polymerase binds the promoter and starts reading nucleotide sequences, it will have to stop reading at the operator site because that site is blocked. I think of it as being kind of like train tracks. You know how at train tracks around here, the train is going down the track, but when it crosses a street, the arms come down at the crossing and it blocks traffic, right? And the train goes by, but traffic is stopped. So the operator site and the repressor are kind of like the arms that stop the traffic, and the traffic in this case is RNA polymerase. Downstream of the control region, you have what are called the structural genes, and these genes can be different. The structural genes are the genes that are responsible for the operon's function. Okay, the structural genes are the ones that are responsible for the operon's function. All right, so I know this is new. Let's take a look at an example. Inducible operons are operons that are off. That means they're not expressed, but they can be induced. That is, under the right circumstances, the genes can be turned on and expressed. I think about it kind of like, pregnant women when they reach term and it's time for them to have a baby, but the uterus is very quiet and there are little or no contractions. A doctor may give an inducer to make contractions in the uterus increase to aid the birth of the child. So there's no activity, but then you give this chemical and suddenly we increase how much activity is happening in the uterus. Here, these genes, the structural genes of an inducible operon, are quiet. But if we give the right chemical, okay, then we get activity from these genes. So that's what we mean by inducible. So the lac operon is kind of the classic example of an inducible operon. Here we have the control region with the promoter and operator sites that you're already familiar with. And then down here we have three structural genes. The first one is called beta-galactosidase. It's LAC-Z, is the name of the gene, but it encodes beta-galactosidase. This is an enzyme that breaks down lactose to release glucose and galactose. The glucose can be immediately used by the cell for energy, and then galactose can be broken down for energy as well. So that's the Z. The Y is beta-galactoside permease. This is an enzyme that allows lactose to come into the cell. And then LAC-A down here on the end, we don't really know what it does. It's beta-galactoside transacetylase, but its role isn't really clear. So that's why I have a question mark there. Don't worry, you don't have to remember the exact genes. What you need to remember is that the structural genes are related to function. Ordinarily, the cell doesn't need to break down lactose, so this operon is typically off. So, as I mentioned before, there's a gene that encodes the repressor protein that sits just outside of the operon. That's the gene I that you see here. That gets transcribed, producing a repressor messenger RNA, and then when that's translated, we get a protein that has a specific three-dimensional shape. And this shape is designed to recognize the nucleotide sequence of the operator site. So the protein binds the operator site. When RNA polymerase binds the promoter, which is just beside the operator, and it tries to open up the DNA and start reading nucleotides, it's got to go past the operator site to start transcribing the structural genes down here. With the repressor sitting there, however, RNA polymerase can't move past the operator, so transcription is blocked, and these genes down here, the structural genes, do not get expressed. Now, in the instance where there's a little bit of lactose in the environment, lactose can act as an inducer. So what it does is it is able to bind the repressor protein and change the repressor protein's shape. In this particular three-dimensional shape, the repressor can no longer bind the operator, which means the operator site will be empty. So when RNA polymerase binds the promoter, it can open up the DNA, start reading nucleotides, move past the operator site, and transcribe the structural genes, leading to the production of an operon messenger RNA. Then this operon messenger RNA gets translated by ribosomes to make proteins that the cell needs to metabolize lactose. So lactose is the chemical that initiates activity of the structural genes. It is the inducer, and that's why this is called an inducible operon. The inducer binds the repressor and prevents the repressor from binding the operator. That turns on expression of the structural genes from this operon. Now let's look at catabolite repression because it's somewhat related. Let's say you take an E. coli cell and you put it in media that has glucose. If you track the growth of the cells over time in glucose medium only, you get a growth curve that looks like this. On the other hand, if you do the exact same experiment, but instead of having glucose in the medium, you have lactose, you get a growth curve that looks like this. So they look kind of different, right? Part of the reason why the growth curve looks like this is that there's more enzymes that are involved in metabolizing lactose than there are in glucose. So that means that growth is going to happen more slowly. Now, here's the interesting thing. If you take E. coli and you put it in a growth medium that has glucose as well as lactose, we're going to see that the growth of the E. coli initially looks very similar to what we saw over here when only glucose was around. After the bacteria use up all the glucose, we'll see the culture enters a lag period. Do you remember what lag means from chapter 4? Or rather from our uh, microbial growth chapter? Right. Remember during a lag period, the cells are detecting what nutrients are in the environment and they're adjusting their metabolism. They're adjusting their gene expression so that they can metabolize whatever nutrients are around. So we see that happen, and then we see the cells start metabolizing lactose because the growth curve starts to take on the slope that we see with lactose. So they use up the glucose first, then they make adjustments so that they can use lactose. And one thing that I hope you'll remember is that there are many, many, many bacteria that prefer to use glucose over other carbohydrates for energy. And part of this is probably because we know that glucose is an ancient biochemical pathway. Some very, very old cells probably developed glycolysis so that they can break down glucose to metabolize ATP. The reason we think that's so is because many, many, many cells, most cells, have the ability to break down glucose. Glucose is quick energy, so the cells prefer to use that first, and then they'll switch to something else if they need to. But how does this happen? It's really interesting. So the first thing that we'll notice is for cells that do catabolite repression, their promoter is a little bit different than other promoters. And the reason for that is we have the standard sequence where RNA polymerase will bind to transcribe the genes that are part of this operon. But there's also this sequence nearby, a CAP sequence. So CAP stands for catabolic activator protein. This is another protein that binds at the promoter and it helps RNA polymerase bind the promoter in the presence of a specific molecule called cyclic AMP. Cyclic AMP is called an alarmone. You can think of it as being an alarm hormone. So what happens is cells start out metabolizing glucose, and after they've used up all the glucose, they start producing lots of cyclic AMP as kind of a hangry molecule. This is a molecule that gets produced when a cell is under stress. So when cyclic AMP is bound, or sorry, when cyclic AMP is made, then it binds CAP protein that's in the cell and activates that protein. Let's take a look. So in this figure, down here is when glucose is high, so there's lots of glucose in the environment, and up here is when glucose is low. There's not very much in the environment. Let's look at what happens when glucose is high first. So when there's lots of glucose in the cell, remember I told you the cell wants to use that glucose for energy. So there might be a little bit of cyclic AMP around because it is a signaling molecule for lots of pathways in the cell, but there's not very much. And because there's not a lot of cyclic AMP around, and what cyclic AMP is around is being used for other pathways, that means that the CAP protein remains in an inactive form. It doesn't bind its sequence in the promoter very tightly, which means that RNA polymerase doesn't bind the promoter very tightly, and there's very little to no transcription of the structural genes. And that's fine because the cell doesn't need the structural genes for breaking down lactose in this case. But when the level of glucose gets low, the cell starts panicking. It produces a lot more of the cyclic AMP, and the chances of that cyclic AMP binding the CAP protein go up. When cyclic AMP binds the CAP protein, it activates it. And in this form, the CAP protein can bind the promoter region of this catabolite repression operon. Remember, CAP protein helps RNA polymerase bind its promoter so that transcription can occur. So that's exactly what happens. We start getting transcription of these structural genes. And remember, in order for this to happen, the cell has to be starving, but there also has to be lactose around, right? So if there's lactose around, then that means that the repressor protein cannot be in the operator site, so transcription happens. If the cell starts panicking like this and there's no lactose around, transcription of these genes still won't happen because the repressor will be in the operator site. All right, so how does the cap protein work? This is a molecular model showing what happens. The red that you see here, you can almost see that it's a double-stranded molecule. That's DNA. The blue molecules up here are the cap protein, okay, cap protein monomers. And then you see this pink here in the middle? That's the cyclic AMP that activates the cap protein so it can bind the DNA at the promoter. What it actually does is it bends the DNA, making it easier for RNA polymerase to bind the DNA and transcribe. So I've kind of explained what happens right now. Let's fill out this question here at the top of this slide to see if you kind of get what's going on. I will admit the operon is one of those things that people sometimes need to hear a couple of times in order to understand it. So you may need to play this video more than one time. Also, don't forget, I'm available during office hours, by email, and by text to answer any questions that you might have about this. All right, let's do the question. When glucose is, let's say, high, so when there's lots of glucose in the environment, okay, the answers for these questions will be either high or low. So when glucose is high, cyclic AMP is, choose one, high or low? Low. Remember, cyclic AMP is a stress hormone, a alarmone that the cell produces when it's starving. So when there's lots of glucose, the cell's not starving, so we don't need a lot of cyclic AMP. How about lac transcription? Lac transcription will also be low. Why? Because without cyclic AMP, the cap protein doesn't bind the promoter. And if the cap protein doesn't bind its promoter site, then RNA polymerase can't bind the promoter site. And then we get very little transcription. How about this one? When glucose is low, cyclic AMP is high. That's right. How about lac transcription? Right. That's also high because now the cap protein is bound to the promoter, which means RNA polymerase can bind, and as long as lactose is around, we will get transcription of the structural genes. Good job. So let's talk about the last operon. This is called the repressible operon. And you know, when we talk about repression, we're basically talking about holding things back. I could think about a time when I was really, really angry with my sister and we were having an argument. And it took everything within me to not say what I was really thinking and what I wanted to say. I had to repress my feelings and my anger so as not to hurt her and so as not to essentially pour gasoline on the flame that was our argument. And I'm grateful that I did that, but I had to repress. I had to hold back what I was thinking and what I wanted to say. So in a repressible operon, the cell holds back expression of structural genes so that it doesn't produce molecules that the cell doesn't need. Kind of the best example of an operon that's repressible is the tryptophan operon. Tryptophan is an essential amino acid for humans, meaning we have to eat food and absorb it from our food. We can't synthesize it. You've probably heard about tryptophan because for a while people said that eating turkey at Thanksgiving released lots of tryptophan, and that's what makes us all sleepy after the Thanksgiving meal. So this is that same amino acid. Bacteria can synthesize their own tryptophan. They don't have to rely on absorbing it from the environment. So this is the tryptophan operon. There are 5 genes that encode proteins that synthesize tryptophan. This is what the operon normally looks like. You can see that there is transcription from this regulatory gene, which results in the production of a repressor protein. But in this shape, the repressor cannot sit in the operator site. Since the repressor is not here in the operator site, that means that RNA polymerase can bind the promoter, open it up, start reading nucleotides, and start transcribing the structural genes for the tryptophan operon. The result is the production of an operon messenger RNA, and then proteins get synthesized from this RNA. But imagine for a second that a bacterium finds itself near a dead animal, and that animal is decomposing, releasing proteins into the environment. The bacterium is going to produce its own enzymes that it'll secrete into the environment. Those enzymes will break down the proteins, releasing amino acids, and then the bacterium will absorb those amino acids, one of which can be tryptophan. So now the cell finds itself in a position where it's synthesizing tryptophan, but it's also absorbing tryptophan. So that's kind of wasteful. What the cell will do is it will turn off expression of tryptophan synthesis as long as there's tryptophan from the environment around. So here's how that happens. We get transcription of our repressor from this regulatory gene. In this particular shape, the repressor doesn't bind the operator. However, when there's tryptophan in the cell, that tryptophan can bind the repressor and change its shape. In this new shape, the repressor can bind the operator site here, and while the repressor is in the operator site, that means that RNA polymerase cannot transcribe the structural genes because it's blocked by the repressor. This turns off expression of those genes. As long as there's a lot of tryptophan in the cell, then this is exactly what happens. The tryptophan binds the repressor and keeps the repressor in this active shape. As the cell starts using up the tryptophan to make proteins, there'll be less and less free tryptophan in the cytoplasm, which means there'll be less and less tryptophan to bind the repressor and keep the repressor in this shape. So gradually what we'll see is a shift in shape back to the inactive form. And as that happens, that means that the repressor won't be in the operator site and will slowly get transcription of these structural genes and we'll get synthesis of tryptophan again. We call tryptophan a co-repressor in this case because it is responsible for repressing gene expression along with the repressor. So that's it for this video. I've talked about constitutive versus non-constitutive gene expression. Remember, constitutive genes are expressed at a fixed rate, but they're expressed constantly. Non-constitutive genes can be turned on or turned off based upon what's happening around a cell. We also talked about the parts of an operon and their functions. Importantly, you'll want to remember that the promoter is the site where RNA polymerase binds the operon DNA and will start transcription. You'll want to remember the operator site is where the repressor protein, which turns off transcription, can bind. And you'll want to remember that the structural genes are the genes that are related to the function of the operon. You'll want to be sure you understand how repressible, inducible, and catabolite repression operons work. That will be definitely a question on the exam, and a good way to practice would be to work through the practice problems on the practice exam. It will walk you through the activity of an operon and ask you to use the knowledge that you have of operons to answer questions. So that's it for this video. In the next video, we're going to look at horizontal or lateral gene transfer. I'll see you there.